Yeah, this is the extended presentation. Since last month, I gave related to that regenerative medicine, self-free regenerative medicine. So here, I want to talk about the adipose-derived stem cell and secretome and their application, especially for the musculoskeletal diseases. So, as we know that regenerative medicines and their clinical readiness have so many plus and minus, as well as some practices is still going on. So first of all, we have to know that cause and cure, the regenerative impacts, and catalyzing the new pathway, addressing, you can say, unmet need, and then workforce readiness, that educated proficiency. So under this framework, you can say finding the regenerative medicine, and our lab is mainly focusing on cell-free regenerative medicine development. Though in regeneration, basically what we are doing, we are making, you can say, our idea is that to bring the homeostasis. Homeostasis can bring by two way, either replacement of, you can say, a new cell to the tissue, extracellular matrix, which can regenerate to the entire desirable tissue and attain the that tissue or organ in a homeostatic, you can say, condition. The second one, we can reprogram the resident cell, and that cell has to be provide the homeostatic condition which is required to maintain the regeneration of the tissue or system. So our hypothesis is that we have to reprogram the resident cells and using the certain type of the secretome. So, however, till date, you can say cell-free regenerative medicine have acellular, that extracellular vesicles, gene therapy, parakine, that is growth factor and cytokine, and also some, you can say, modified mRNA, microRNAs, and small molecules or chemicals are in a practice to reprogram the cellular system or tissue, or as well as, you can say, the resident cell to achieve this one. There is some other, you can say, engineered material, that is scaffold, hydrogels, or cell seat is there, by which we can also reprogram the, you can say, resident cells and to achieve the homeostasis. Today, I'm not going to talk about stem cell, but here the topic which we decided to, you can say, discuss, that is the stem cell derived exosome. So secretome. So when I say secretome, so there are so many things, but our major focus is exosome because it's well-defined, you can say, structure which we can use for the therapy process. So in case of the stem cell, we know that the mesenchymal stem cell is among the mesenchymal stem cell, that adipocyte is our target. So adipocyte is very important and abundant type of the, you can say, tissue which is available in our body, and we can get as much as we want. So obviously getting secretome from the adipocyte is much higher, you can say, possibility to get the secretome from the adipocyte is much higher than any other type of the stem cell. So here we have to understand then what, as per definition, that what is adipose-derived stem cell. We know that adipose-derived stem cells are mesenchymal stem cells that are obtained from the abundant, you can say, adipose tissue that are on plastic, you can say, culture flask, can be expanded into in vitro and have the capacity to differentiate into multiple cell lineage. Unlike bone marrow derived that mesenchymal stem cell, adipose stem cell can be obtained from the abundant adipose tissue by minimally invasive procedure, which result in high number of the cells. So that's why it is very, you can say, having lot of advantage, and that's why researchers are very much hypnotized with this adipose derived stem cell. And therefore, adipose derived stem cell are promising for regenerate the tissue and organ damaged by the injury and disease. But we know that cell therapy have some disadvantages, and among that, most important concern that immunorejection and rejection from extracellular matrix. So these two concerns are very important, and therefore the cell-based therapy have certain limitations also. However, secretome, when we talk about the secretome, then it's very important. You can see in this particular slide that secretome may broadly classify into growth factors, cytokines, and extracellular vesicles. Nowadays, extracellular vesicles, I mean, if you see the research publication, or you can say a trend of the research and the clinical trial, you will be happy to know that within 10 years, this particular extracellular vesicles that get lot of attention from the researchers as well as you can say pharma companies to develop the new type of the pharmaceuticals as well as you can say therapeutic procedure. So in case of the secretome, we can define like paracrine and extracellular vesicle. Paracrine like bioactive molecules such as insulin, like growth factor, or you can say vascular endothelial growth factors, etc. And also cytokine, as I already said. What these secretome are performing in a micro environment. So in micro environment, they play you can say millions of biological function because they have, I mean, by their combination, they perform the different type of the bioactivity. So you may see that this secretome have different type of the you can say protective role, like neuroprotective, as wound healing and angiogenesis and anti-inflammatory as well as anti-apoptotic. And this secretome may contain like miRNA, which is epigenetic regulator. Then it have the TGF, you can say growth factors and the cytokines, and their combination also play a specific type of the, you can say cellular pathway, triggering this as a specific cellular pathway. And then we have the different type of the phenomena. Now come to the growth factor and cytokine. Earlier, it believed that growth factor and cytokines are two different things, but the growth factors are, you can say, signaling molecule that regulate the cell proliferation, differentiation, and migration. However, the cytokine was first used to define as a subset of growth factors and influencing the immune and hemopoietic cells. However, it is now clear that cytokines can act on many different cell type and have diverse effect on proliferation and differentiation. Therefore, cytokine is now used more broadly and you can say changeably with the growth factors. Now come to the growth factors. As a lot of growth factors is well defined for their role in a, you can say in a vital role. For example, like IL-3. So IL-3 enhances the human you can say osteoblast differentiation and bone formation in both in vitro and in vivo condition. If you see that stem cell factor is a kind of the growth factor that exists in both as a membrane bound and you can say as well as you can say soluble form, and it promoting the proliferation, migration, as well as you can say survival and differentiation of the hemopoietic progenitors, melanocytes, and germ cells. Similarly, IL-6 and IL-6 and you can say granulocyte macrophage colony stimulating factor like GM, CSF. These all have, you can say, well documented for their different type of the vital function. And TGF-beta is one of the very important, you can say, that growth factor, which TGF-beta signaling, you can say, additionally control the expression of that plethora of homeostatic gene whose activity determine the cell proliferation, extracellular matrix production, paracrine factor, secretion, cell contact, immune function, and tissue repair. Therefore, I mean, in a lot of therapy procedure, people are using TGF-beta as a wound healing, as well as you can say that healing medicine, or you can say therapeutics. So this is about the you can say basic glimpse about the different type of the growth factor. Similarly, you can see that that FGF, it is involved in angiogenesis, that neural, you can say survival, and synaptogenesis, bone repair, and it means, I mean, whatever growth factor, or you can say cytokine, which we know right now, they all have different type of the vital role. However, these studies say that their combination and their optimization is a very important to achieve the targeted, targeted phenotype like proliferation or differentiation or migration, or say, you can say extracellular matrix remodeling to achieve the homeostasis. Among them, exosomes are very, you can say most celebrated secretome in past 10 years. However, this story of the exosome is not very older, so therefore, is still so many information. I mean, in a development phase, and people are looking for some more information about precise biomechanism, or you can say guided secretion, as well as you can say targeted secretion of the exosome to achieve the desirable function. So in 1983, that it was coined as a vesicle that contribute mammalian, you can say, reticulocyte differentiation and maturation. So exosome as one of the, you can say, three subclass of extracellular vesicle, and are shaped through the budding as a intraluminal vesicle. Within the luminal space of late endosome or multivascular bodies, through endosomal complex required for the transport that dependent or ESCART dependent pathway. In case of the, you can say, multivascular bodies include endocyte marker, and it compasses the RAPs and lysosome associated membrane glycoprotein, which make them distinguishable from the autophagic bodies. So we can easily distinguish this exosome from the different other bodies lysosomal associated, or you can say other autophagic bodies. So exosomes are responsible for the range of the cell-to-cell communication, you can say, axis allied with the several physiological and pathological activities. And in case of the this uh, multivascular bodies that comprising a high ratio of the cholesterol could merge with the cellular membrane or B cell and release the exosome. So exosome production including the hypoxia, overexpression of the tetraspatine CD9, along with the overexpressing hypoxia inducible factor alpha. And in case of that was the normal characteristics of exosome. However, when we talk about the adipose derived stem cell exosome, so adipose tissue is an endocrine organ which involved in management of the energy metabolism via secretion of the epidokines, hormones, and recently described secretory microvesicle, that is exosomes. So exosomes are rich in possible biologically active factors such as proteins, lipid, and RNA. This secretory functions of adipose tissues is affected by pathological process, and exosome are, you can say, nanoscale membrane vesicles secreted by donor cell and transfer to the you can say genetic information to the recipient cell, where the encapsulated nucleic acid and proteins are released. Moreover, the potential significance of the exosome as a therapeutic vesicle or drug is also, you can say, discussed in so many papers, and here in this lecture we will also discuss about that. So this is the, you can say, schematic diagram demonstration for the exosome development. You can see easily that this is the early endosome, then we will have the multivascular body, and this multivascular body, it will make the late endosome, and obviously it will be released this kind of the microvessels, and these microvessels basically contain protein, lipid, and so many, I mean, you can say RNA, I mean nucleic acids, and etc. And because this particular bodies that go to the recipient cell and allow, you can say, to release the certain kind of the information, as well as control the biological pathways to inform the cell that how to behave, I mean, in a different way. So this is the basic, you can say, idea about the exosome. But important thing is that because why exosome is so important. So when we see that exosome structure and it's a content, so exosome involves, you can say, numerous cytoplasmic and membrane protein, including receptor, enzymes, transcription factor, extracellular matrix protein, nucleic acid, like mtDNA and ssDNA, like single stranded DNA, or you can say double stranded DNA, that mRNA and miRNAs, etc. And also lipid. So although the cell addition molecule, integrins, and you can say tetraspanin, and major histocompatibility complex protein are mutual, that amongst various varied exosome fusion and transferring associated proteins such as RAP2, RAP7, others are also there. And you can say exosomes in closing, you can tetra spanning 8, and it's related to CD49, these shapes. And you can say interaction with the endothelial cells by CD106 and ligand for the CD49D is there. So it means the endosome is not, just a general body. It have the specific, you can say, receptor and ligand type of the interaction to the target cell. And here it's a pictorial diagram about the exosome structure. However, this particular is just a pictorial diagram, and you can see that it have like tetra spanins, and it have the cholesterol, because high cholesterol promote, you can say, easy binding with the plasma membrane to the other cell. Then it have microRNAs, and these microRNA play a lot of epigenetic regulation of the different type of the pathway, because it can regulate the protein synthesis by mRNA regulation after binding with the mRNA, and the mRNA get, you can say, denatured, digested by the RNAs. It also have DNA, it have some certain cellular protein and other proteins also it have, and it also contains chaperone, like you can say chaperones also is there. So obviously it also help in your protein folding of the target cells. So there are so many type of the vital functions it can perform. And this is just like, like a perfect engineer who can fix the target cell and reprogram the target cell for the specific function. So here, the formation and release of the exosome, you can easily see that the receptor-mediated endocytosis is there. And once the endocytosis has been performed, then they make the early endosome. This early endosome, by the Rab9, it makes the multivascular body. And after that, it have the certain type of the Rabs. And these Rabs will allow to release the, you can say, exosome to out. And here you can understand that how this exosome can get inserted into or release their content into the desirable cell. So you may see that how they are behaving. Like, I mean, first one is fusion, then another one is your juxtacrine signaling, and third one is your phagocytosis. So all these three processes possible. So by that it can interact. Like membrane fusion allow for the transfer of the both internal cargo molecule and membrane protein by merging with the plasma membrane to the target cell. And similarly, you can see that juxtacrine signaling activate the intracellular signal pathway through the ligation of the target cell membrane receptor with endosome membrane ligand. In case of the phagocytosis, it's a well-known process that allow to, you can see, sterilization of the whole exosome by target cell, and it allow for the lysosomal, you can say, degradation of exosome. That biological effect of exosome action depend on type of interaction with the target cell. So this is the mechanism, and here this is very important kind of the that whatever mechanism I described, it's a much more you can say explained way, that how it is going to bind with the receptor, and after binding receptor, this go inside the cell, and the target cell perform you can say different type of the genetic expression based on the signals, or you can say molecule which guide to the target cell. Now come to the important aspect why we are talking about the exosome. So exosome is, as I say, that it has so many, you can say, guiding type of the molecule and phenomena. And exosome isolation is very important, and character of the exosome is also defined by so many different type of the extraction methods that also you can say influence the final role of the vital role of the exosome. So here some already established, you can say, methods are there, like ultracentrifugation, like density gradient centrifugation, size exclusion chromatography, filtration and immunoaffinity capture, like microfluidic technology, magnetic bead immunoassay, then kit isolation method, and polymeric precipitation is there. So and you can say they all will become as an advantage and disadvantage. So the disadvantage is, I mean, it's still because it's most of the exosome related research are under preclinical trial or some clinical trial. So once it has been completely understood about the clinical you can say use of the exosome. So at the moment, I mean, after engineering, disadvantages can be you can say overcome all the disadvantages. So this is the different type of the isolation method, and in our laboratory we are performing the density gradient centrifugation, not other method. So the question from where we can isolate the exosome, we can get exosome from any you can say body part, including the CSF, saliva, milk, blood, urine. However, in our system we are trying to isolate the exosome from our cell culture media. So here what we do, I mean, first we take the sample, and the sample should be, you can say, that analyzed by ultracentrifugation, or you can say size exclusion chromatography or commercial kit. Commercial kit is very expensive, so most of the time for academic research we are using ultracentrifugation. Basically this ultracentrifugation is density gradient centrifugation, and then we have the exosome. This exosome is further characterized, as I already say, that exosome have certain kind of the markers. So these markers can be easily characterized using immunoblotting, or that electron microscopy is also there to know that uniformity of the exosome. And another that is the flow cytometric analysis is one of the important tool to analyze that. So in general, when we are trying to characterize the exosomes, exosomes are usually, you can say, 30 to 100 nanometer microvessel. And obviously the marker which is present over there, that's CD63 and TGS101, that helix and flotilin, then its density is a little bit higher than the water, and it consists of the protein, lipid, and different RNA species and DNA. It determined by the control content, cellular origin, and physiological state of the cells. So obviously, as I already say, that we can tune the different type of the exosome secretion by providing the specific condition to the culture media, and that we are optimizing in our laboratory also. So this is the molecular, you can say, markers by which we can identify our desirable exosome. For example, that MSC derived exosome. Here we are mainly focused with the MSC derived. Among that, adipose derived MSC is our major target. And in case of the adipose derived MSC exosome, the CD9, CD34, CD44, CD68, CD81, CD90, and CD105 are the you can say markers present over there. And as per characteristic miRNA, it have miRNA 155 and 146. And if you see the cargo, or you can say pathway which is directly affected, so you can see that the P13K or AKT or that TOR or TGF beta, and so many pathways can be affected easily. And here the role is immunosuppressive. So this is the basic information about the exosome from the adipose derived. However, the other cells also produce exosome, and their marker is varied because of the cell type. And once we see the physiological role of adipose derived, you can say, exosome, the homeostasis of the body is the result of the interaction between the various tissue. And as the largest, you can say, secretory organ, adipose tissue release various hormone and adipokine, which I already discussed earlier. And this increase, you can say, enlarge the adipose tissue to obsess the patient, can cause the various metabolic and cardiovascular complications. Recent study identify adipose tissue derived exosome as a type of the intracellular communicator mediator between the adipose tissue, and other can perform lot of vital role. And you just see this particular picture. I hope it's very much clear to all of you that this is our you can say adipose stem cell. And this adipose cells, if suppose we are looking one function exosome containing miRNA 4505-5P, it can provide that circulatory system. It can also provide the atherosclerosis and adipose formation is there. then it also have the placenta function, fetal growth, and also you can see that endocrine system, it can help over there using the certain type of the pathway via the miRNAs. And it can also affect the M2 macrophages to M1 to M2 macrophages. So adipose tissue derived exosome in lipid metabolism, their involvement in you can say regulation of insulin resistance, as well as they are highly involved in that regulation of inflammatory immunity, and it is also involved in other system like neuroprotective and etc. The exosome, you can say, improve the endothelial function, and also exosome treatment for the metabolic syndrome is possible. So here that adipose-derived exosome in the regulation of the biological process, which is already documented by several researchers, like that insulin resistance, cell proliferation, then colitis aggregation, and lipid, you can say, metabolism, immune homeostasis, you can say, homeostasis inhibition, etc. And that also perform the M2 polarization, and we can see the different type of the biological role in our body. And here you can see that therapeutic application of adipose derived cells here, that is and it can protect, I mean, diabetic nephropathy, you can say angiogenesis would be there, and also cardio protective and hepatoprotective, as well as you can say it provide anti-inflammatory effect. So adipose derived tissue is exosome is there. Now the therapeutic effect, which is already validated, that exosome derived, these all are the exosome derived, and like native exosome, it promote the wound healing, and also it improve the erectile dysfunction, then promote angiogenesis and etc. Now come to the specific role here in our you can say laboratory. We are looking for some different aspect, like exosome as an anti-cancer. In case of the cancer, we know that any cell with, I mean, cancer therapy involves so many things, like from the cancer development to the apoptosis is involved over there, and exosome have potential to induce the apoptosis as well as reduce the inflammation, and also it can modulate the M2 polarization. So these all phenomena can be possible, and if we are able to reprogram the entire tumor microenvironment using the exosome, then we can easily find out a new way to treat the you can say non-malignant tumor. Here, this is the mechanism by which, I mean, it is producing, you can say, different type of the molecule. And if you see that the signal transduction pathway of exosome in tumor microenvironment, the most important cell in tumor microenvironment mainly include that CAF and CSC and MSC, etc. These four type of the cells use exosome to promote EMT tumor metastasis and drug resistance through the variety of mechanism. So if we understand that how to you can say control this particular exosome, then obviously we can overcome this drug resistance to the tumor metastasis. Obviously, I mean, the study is going on, so maybe in future we will have much more insight information. In case of the exosome mediated, you can say that a tumor, so we can see that the cancer cell, that must be our target, and we can deliver the exosome, which includes certain type of the drug, as well as you can say miRNA, which can easily regulate over there. So that's exosome engineering is now very popular for as a drug carrier, and if because it have the ligand, so it can directly bind with the specific tumor and release the drug over there. So, and as you know that we are working in orthopedic department, so healing is very most important study criteria for us. And here that exosome also contain, especially from the mesenchymal adipose derived mesenchymal stem cell, it has so many type of the you can say healing promoting miRNA, which can be promote the healing. As you know that the healing, we have three phases, that anti-inflammatory, then we have the proliferative and differentiation phase, and finally that remodeling phase. So the remodeling is very important for wound healing, and exosome has been already identified their role in remodeling of the wound. So you may see that the pathways which can easily regulated by the exosome during the healing. So major pathways, that is the like growth factor, and they involve in VNT pathway, IGF pathway, and utrophin and TGF beta, VEGF, and other pathways are there. So obviously it can promote the proliferation as well as angiogenesis, and finally that can also fibroblast proliferation, extracellular matrix deposition, and collagen synthesis is there. So this collagen synthesis is very important for extracellular matrix, you can say, remodeling. And here the you can say schematic diagram showing that major factors which involve in matrix metalloprotease regulation released by the exosome, and you can say the changes in the tendon that occur adding the healing process. So you just see this, you can say the blue boxes that include the critical mediator for the normal healing tissue remodeling. An exosome release matrix metalloprotease and tissue inhibition metalloprotease. So yes, this kind of the reprogramming ability of the exosome provide the matrix remodeling for the healing. So in totality, we can say that the exosome ability to, exosome can perform the 4 directional you can say vital activities. So it can promote angiogenesis, it can promote the cell proliferation and migration, and also it can polarize the M1 to M2, and finally that it can remodel the matrix by synthesis of the collagen, and obviously removal of the unnecessary or you can say broken collagen over there. So here, this is the pictorial diagram about how this healing is performed in a tendon like a tissue. So its cell migration should be there, then division and secretion is there. So proliferative and secretion of the collagen is there. By that, it can repair the entire tendon itself. Now the potentials of exosome, as I say that the adipose derived, you can say stem cell have ability to provide all kind of the lineage. A different type of the cell line can be directed from the adipose derived cell. It means it has certain type of the signal capsule like exosome, which can help during the differentiation process. Converting the adipose derived stem cell to the specific stem cell. And this can also provide the different type of the role in a body, and it most of the not most all the here given that has been properly documented and validated by various researchers. So like inhibition of the inflammatory response by activation of the SIP-SK1 and SIP-R1 axis. Similarly, you can see that it's suppression of the Wnt beta catechin pathway, and by that it can protect the liver also. In case of the bone, we are mainly interested in bone and you can say ligament and muscles. So induction of extracellular matrix regeneration by promoting collagen type 2 and synthesis and inhibition of the ADAMTS5. Then promotion of cartilage homeostasis by suppressing Wnt5A. Inhibition of the fibroblast, like you can say synovocyte motility. So yes, this exosome have potential, but as I say, is the exosome can be used for the different type of the studies. So here the table already summarized that the source and cargoes, loading, mechanism, and effect that has been already observed during the study. Like mesenchymal or adipose derived, so it have this much of the miRNA, and each and specific miRNA and the signal molecule siRNA have their specific role, like cell migration, self renewal, you can say reduction of the progression or metastasis, like miRNA 146, etc. As I say that we are mainly focused for the musculoskeletal disorder, and we found that the exosomes, you can say, already studied for osteoarthritis and osteoporosis and except cartilage defect, everything. So in my opinion, that exosome have potential to do lot of things related to you can say healing effect. And here, this is the list. If someone wanna, you can say, more insight about the how this is working. So you may see that how this is going on. And last but not least, is exosome in a drug delivery. As I already say that exosome have ability to target their cell, and if we you can say if we add a drug inside the lumen, so obviously it can directly deliver to the target cell. And so many system is already going on, like engineered exosome as a drug delivery system, which study is already performed. So for example, IRGDA peptide, that receptor is LAMP2B, and for the breast cancer it has been targeting delivered to the dogs and effectively inhibit the tumor growth. So so many you can say trials are going on to with the exosome as a carrier for different type of the disease, and in future maybe after optimization it could be the better tool. Here, this is the mechanism by which we can target to the cell. One is we can add the self-expressing plasmid, which can directly go to the cell, receptor cell, and then in receptor cell, you start production of the desirable protein, which can perform the specific function. The second one is that we can add here the drug inside the exosome and it go and bind to the receptor cell. And third one, we can add the magnetized, you can say, signal. So we can guide the exosome by external magnetic force, and it will reach to the specific organ or tissue to perform the specific activity. So physically and chemically modified exosomes for targeting drug delivery, that exosome with SPION, and it's a magnetic field tumor site. Liver cancer is very, it has been already demonstrated. Then SIRP alpha like CD47, it's a macrophage and tumor cell. So, and similarly, you can go with the hydrophilic cholesterol coupled with the RNA aptamer. So it go to the RNA ligand receptor, folate receptor, and it target the cancer cell and target delivery of the siRNA and miRNA to inhibit the cancer propagation, or you can say to induce the apoptosis in a cancer tumor. So this is you can say like some other secretome which has been already in clinical phase, like EGF, TGF beta, FGF, and PDGF, etc. is in a clinical outcome, and their outcome is already reported for the second phase trial. So here it's a comparison between the stem cell and secretome, while that has been already compared last presentation also. So I'm just coming here that why and how this exosome can help us as a therapeutics development. That first of all, we have to consider like a main limitation in a clinical application of the adipose tissue derived exosome, that remain the resource and quality control among the different batches. That is the biggest challenge with us. Then isolation of the sufficient exosome from the adipose tissue, or you can say establishment of the alternative resource of the fundamental for the potential clinical application. Then techniques to ensure the exosome homogeneity during the biogenesis and isolation and quality control are also wide needed. And the challenges you can say could be solved, that fundamental procedure of the exosome biogenesis and regulation are in future clarified. So yes, I mean, this is the matter of engineering. Once we know the entire, you can say, biology and biochemistry, and obviously omics from the exosome, then we can also engineer for production and quality optimization of exosome for development of the therapeutics for the future. So at current stage exosome studies, we do not have complete, you can say, understanding of the potential therapeutic applicability, and therefore we need more and more study to understand the exact biological condition and how we can, you can say, tune the biogenesis as well as overexpress the specific ligand for the receptor. So but anyway, we have to believe that with continuous research work in the near future, we may, you can say, full use the advantage of the exosome as a natural carrier and bypass the shortcomings. And great progress, you can say, we made in the therapeutics development strategy based on the exosome in a great goodness of vast cancer patient. So this is all about exosome, which we are doing in our laboratory. And here we also did some study related to adipose-derived exosome and their role in degenerative tendinopathy, especially that Achilles tendinopathy. And hopefully in coming month, we will have some good, you can say, outcome from that study also. So I must say thanks to all of you for listening this presentation. If you have any query or question, please allow me to satisfy yourself.
Thank you for your presentation, Vivek. Is there any question or comment? I think the exosome is a very good treatment modality. It sounds like almighty, but the disadvantage of exosome will certainly exist, I think. What do you think is the most disadvantage of exosome uses?
As sir, I already make the advantage and disadvantage of the, you can say, comparison. So I want to explain one more time that in case of the advantage, that therapeutic, you can say, component of the secretome could be customized by modifying the culture condition, which is our plan in our laboratory also. It can cross any kind of the barrier like blood-brain barrier, and can circulate and home to the target tissue because it have the ligand for their specific receptor for the cell, target cell. It have the low risk of the mutation because it don't have, long nucleic acid. So obviously the chances of the mutation is very, very minimum. And obviously it don't have any risk of the carcinogenicity because it don't contain any entire cell. So obviously the mutation or carcinogenesis is not over there. It can bypass the immunogenic type of the response because it is not a whole cell. So obviously it can pass the immunogenic responses. And most important, they are not a living cell. So dealing with them, it's much more easier. In case of the lower risk of the vascular obstruction, even any kind of the, you can say, barrier, as I already said, it can pass. So it can reach to the deep of the tissue. In our study, we are focusing for the tendon, and we know that the tendon is the least vascularized connective tissue of our body, and where exosome can easily deliver by local application. So it's a smaller in size that help to penetrate any kind of the barrier. It's easier to store as compared to other regenerative medicine like the cellular system. So it's a very easy to store. We can store like other biologics. And cell, you can say, degeneration or senescence in the host after transplantation is not a concern, because most of the time when we go with the stem cell therapy, we know that the cell degeneration and senescence is a very you can say obvious phenomena which is observed during the stem cell. Similarly, if you go with the, you can say, disadvantages, yes, it's still because this is not, you can say, very old and well established technology. So therefore it cannot be as a use as a cell replacement therapy, because it's totally dependent on the number of resident cells. If the resident cells is less than the critical value, then definitely it cannot induce the proliferation and differentiation. And another thing is that lack of understanding on the mechanism of action. This is not a big disadvantage because till date we have limited knowledge. So obviously maybe in future, after, you can say, having lot of knowledge, this disadvantage will definitely overcome. And lack of long-term safety data, as I already say, this is a very new science, so we don't have much, engineering about the long-term safety, long-term storage. And finally, that is a lack of industry standardization. But lot of, researchers, they are trying to optimize the culture condition to get the single type of exosome or targeted exosome production, etc. But still, that is a matter of research. Then short half-life in the body. Of course, this is a disadvantage as well as advantage. If something have the long, you can say half-life in a body, so it will persist for the longer period also. And the half-life can be tuned also by surface engineering of the exosome, and a lot of researcher has already proved about the how to engineer the surface. Then difficult to purify the specific therapeutic component. But nowadays we know that different type of the surface protein is there. So affinity column is, is already prepared. Though this technology is very expensive because it's only research purpose. So most of the companies, they are asking huge money. But once it become a therapeutic, so obviously a lot of competitor will come in the market for production and purification. So the cost of the purification and type of the technology will help to get the highly purified exosome in the future. So this is all about the advantage and disadvantage associated with the exosome based therapy development.
Thank you. What do you think is the most disadvantage?
So most disadvantage is limited knowledge about the exosome biogenesis and its mechanism.
Okay. Is there any question or comment? 네, 더 이상 질문이나 코멘트 없으시면 이것으로 오늘 미팅을 마치도록 하겠습니다. Thank you for your presentation, Vivek.
Thank you,
Let's
sir.
wrap up today's meeting. Bye.
Bye, sir.